So today I will primarily talk about my PhD research on the origins of homochirality as it's relevant to the origin of life. I'll first introduce the problem and then the mechanistic aspects of how we break the chiral symmetry, and then some experiments that I did in the last few years and some unpublished stuff that is related to geochemistry and rock magnetism with the groups here of Nick Toska and Rich Harrison. And then I will talk about a scheme which we call a central dogma after Francis Crick of Cambridge University, which describes how chiral information spreads in prebiotic chemistry. So what is homochirality? Well, chirality is a geometric property, and objects are said to be chiral if their mirror images are not superimposable on each other, just like our hands. And it turns out that many biologically relevant molecules are actually chiral. And they can exist in mirror symmetrical pairs as left-handed and right-handed objects. But the interesting thing is that biology is very picky when it comes to chirality. It selectively uses left-handed amino acids and right-handed sugars. So essentially, life is a homochiral entity. And that's the puzzle, right? Like what gave rise to this feature of life on Earth? And well, that's a feature of life on Earth, but also it's a prerequisite for life to start, because when we think of origins of life, we kind of think about a genetic code, one form or the other. And something like RNA is composed of chiral units, ribonucleotides, which exist also in a chiral helical geometry. And RNA replicates with Watson-Crick base pairing. And everything basically needs to match such that a strand of RNA can find its perfect Watson-Crick base pair. So essentially, if chirality problem is not figured out, meaning the helices are kind of messed up in their geometrical structure, the pairing of the strands with each other wouldn't be happening. So essentially, the genetic code wouldn't function without, if we don't figure out chirality beforehand. So essentially, we can say that homochirality is not only a feature of life, but also it's a prerequisite for life to start. Because genetic code, something that carries information, is essential for life. So if we kind of think of the origin of life as different stages, in which we kind of first start with the geochemical simple, feedstocks and some energy, which then with some prebiotic chemistry gets converted into biomonomers, which most of them are chiral. And then in the second stage, if we kind of think of the emergence of functional polymers like RNA and peptides and vesicles, like this transition actually wouldn't be realized without we solve homochirality problem. So essentially, homochirality problem is right at the center for us to understand the origins of life. And because of that, justifiably so, like it was recognized as one of the biggest questions in science. In fact, one of the biggest 125 questions, in addition to origins of life in that list. And it's a massive problem, and we can break up into parts such that it's easier to tackle. And it's kind of convenient to consider the problem in the following aspects. So chemically, if we make a synthesis of chiral molecules without an external chiral bias and influence that would kind of force the system, you basically make a racemic mixture, which is a mixture that includes an equal amounts of right-handed and left-handed molecules. And then you need some agent that couples to molecular chirality that would be available on early Earth, which would introduce a bias in some direction and kind of nudge the system and introduce what is called an enantiomeric excess. And then once you kind of break the symmetry by physical or chemical means, you can amplify that to realize homochirality at a compound level. Okay? But that's not enough, because life is not a single molecule thing, right? You need to be able to propagate that homochirality or do this process for every single chiral molecule. So we also need an effective propagation of the so-called chiral information, right, L or D, from the molecule that you attain homochirality at to the rest of the chemistry, such that you start from a completely racemic system and end up with a homochiral network. Okay? And in my talk, I will talk about all aspects of this problem and hopefully try to convince you that we have a viable symmetry breaking mechanism and a way to amplify that small excess and then propagate it to realize systems level homochirality from compound level. Okay? And now first I'll talk about the symmetry breaking part and the mechanism that we use to break the chiral symmetry. But before that, I will talk about the CISS effect because it's at the core of what we're doing. Okay, so the CISS effect is a phenomenon that was recently discovered, like roughly about 10 to 15 years ago, by the group of Ron Naumann in the Weizmann Institute. And the seminal experiment that basically established the phenomena is the following one. So what they do is they take a gold surface and then place a monolayer of DNA molecules on it. And DNA is a chiral molecule, and they put homochiral molecules on this layer. And then they shine light on the surface, and then they eject photoelectrons by photoelectric effect. And because gold is not a ferromagnetic surface, right, it doesn't have any spin bias, so the electrons that are ejected from the surface to begin with are not polarized. But the interesting thing is, as the electrons go through the chiral monolayer, they get heavily filtered based on their spin alignment. Okay? And when they measure the spin polarization properties of the electrons that went through the chiral monolayer, they become heavily spin polarized nearly up to unity. And that was unprecedented at the time because all the calculations you make for closed shell, organic molecules shouldn't give that number. So it's clearly like some new physics, right? And that effect was verified many times with different molecules of different handedness, and it was found that actually the spin alignment was controlled by the handedness of the molecule, in which the chirality strongly couples the electron spin at room temperature. So these are, experiments that were done at room temperature, which is important to emphasize. So a consequence of the CISS effect, and that's what I want you to take away from essentially the whole CISS thing, is the following. So because CISS effect basically suggests that the chiral molecules or the chiral potential of the chiral molecules can act as spin filters, essentially if you polarize a molecule, meaning if you push charge from one end of the molecule to the other one, you basically realize also spin polarization that accompanies charge polarization. Because essentially if you charge polarize a molecule, right, what you do is you move charge inside a chiral potential. And because of the CISS effect, the propagation efficiency of electrons inside a chiral potential is heavily dependent on their spin alignment. Charge poles that are realized when you polarize a molecule will be associated with spin poles as well. And the spin poles will be different, varying for different enantiomers of the same chiral molecule. So what that means is if you charge polarize a chiral molecule, you make the molecule magnetic. Okay? And the molecules, the closed shell chiral organic molecules, are not magnetic when they're not polarized. So this is the main takeaway, and we will use this concept later on and on. So, but you may say, well, this is using chiral molecules to filter out spin, right? But what we want to do is kind of the opposite. We want to use spin, which is a bias factor, to filter out chirality, which starts from an equal mixture. So it's kind of applying the CISS effect in the opposite direction. And where can spin bias come from on early Earth? It can come from magnetized surfaces, because magnetization, by definition, means average spin alignment. And there were a lot of magnetic minerals on early Earth, and I will explain how those will now help us to break chiral symmetry. So imagine a magnetized surface, with its spins aligned, and two molecules approaching the surface. Two molecules of the same, two enantiomers of the same chiral molecule, the left-handed one and the right-handed one. As these molecules approach the surface, they interact with the surface with van der Waals forces, and then they get inductively charge polarized. Okay? And that happens when molecules interact with surfaces or molecules interacting with themselves. That's a mundane phenomenon. And the charge poles that are associated with the molecules as they approach the surface is going to be identical for left and right-handed molecules because the charge motion, in this case, is kind of agnostic to chirality. But what is different for these two systems is the spin poles that are associated with these charge poles. Because just as we discussed, if you polarize chiral molecules, charge polarize, that charge polarization gets accompanied by spin polarization, which will be different for different enantiomers of the molecule. Because let's say for this one, essentially if the upspin moves more effectively, you will end up with the downspin here, and it will be just the opposite one for the other molecule. And once the molecules become magnetic, transiently magnetic near the surface, they will selectively interact with the magnetized surface because, essentially you can think of it as like two magnets pushing each other or pulling each other, right? Like the surface is one pole, and the ends of the molecule are different poles. So one will be pushed away from the surface, and its effective concentration will be decreased. The other will be pulled by the surface, and its effective concentration will be increased. Meaning any surface process that is done with chiral molecules on spin-polarized surfaces will be enantioselective. Okay? And the energy difference between these two configurations is given by the spin exchange energy difference between this triplet-like energy configuration and the singlet-like energy configuration, which is actually much larger than room temperature. And that's the counterintuitive thing about the CISS effect, right? It doesn't come from Zeeman-like weak interactions that you need to apply giant magnetic fields. It's actually a spin effect that relies on kind of the Fermi exclusion principle that is really, really strong at room temperature. So that's the interesting thing. And what this tells us is magnetized surfaces can break the chiral symmetry and function as chiral agents, although they're completely achiral. Like these are iron oxide, for instance, or iron sulfides, which are completely achiral, but because of the CISS effect, they act as chiral agents. So this was one part of it. And as you can tell, this is a surface process, right? Because it requires the kind of the wave function or the electron cloud of the molecule to overlap with the surface. So it is not a very long-range effect that can propagate away from the surface into the bulk of the solution. So how are we going to kind of take care of the amplification that we just discussed? Well, that can come from crystallization, actually. So now I'll tell you how chiral molecules crystallize from their racemic solutions. So typically, if you crystallize a chiral molecule, they crystallize in two different ways. The general way, the more common way of crystallizing chiral molecules is in the state called racemate, in which a distinct crystal chunk is a homogeneous mixture of left and right-handed molecules. So the mixture is overall racemic, as well as it's homogeneously mixed. A little more rare breed of crystals are called conglomerates, which are actually different than what geologists call conglomerates. Apparently, you also have conglomerates, but they're different. And conglomerates, basically, in this configuration, each crystal is individually enantiopure. And roughly 10% of crystals crystallize as conglomerates, according to Cambridge crystallography database. And of course, if you do this process from a racemic mixture, the overall mixture of individually enantiopure crystals is still going to be racemic. But what is happening is if you kind of seed the crystallization at the surface in a chirally selective way, the conglomerate crystallization property of the crystal itself will take care of the rest. So essentially, you will seed it at the surface, and then you will amplify it because the molecules will attract the molecules of the same kind on itself. So essentially, if you combine the surface process on the magnetic surface of a chiral molecule with the conglomerate crystallization, you can kind of nudge the system to go in one direction and then amplify it. So that's what we are going to do, but that's just the mechanistic parts. We had to do this in a molecule that is relevant to prebiotic chemistry, right? It just cannot be any molecule. So now, because of that reason, unfortunately, we need to look at some molecules. And this is a complex system of chemicals, which is called cyanosulfidic chemistry, which was mostly actually discovered here at MRC by the group of John Sutherland. And this is an impressive network of chemicals which essentially starts from hydrogen cyanide, a molecule that was available on prebiotic Earth. And by some series of transformations, it makes shared precursors of amino acids, nucleic acids, and fatty acids. And one interesting line in this is the synthesis of ribonucleotides. And in cyanosulfidic chemistry, pyrimidine ribonucleotides are synthesized from a shared precursor called RAO, ribo-aminooxazoline. And I will look at a more kind of zoomed in version of this synthesis. And here, the synthesis of these family of molecules starts with the first chiral sugar, which is glyceraldehyde. And then as it incubates, as it reacts with 2AO, which is a product of glycolaldehyde as well, another prebiotically relevant molecule, it makes these family of molecules in a racemic state, meaning in a 50-50 mixture of a bunch of different molecules and stereoisomers. And the interesting thing is, one of these molecules of this family is chiral, crystallizes as a conglomerate. So it satisfies that kind of special property of crystallization, which is not very common. It's not very rare, but it's not very common. And it's very insoluble, meaning you can actually crystallize it on a natural environment from relatively low concentrations. And it's pretty stable, meaning if you attain this molecule in its enantiopure form, it doesn't racemize or isomerize in the solution. And most importantly, it's a direct precursor of pyrimidine ribonucleotides. Meaning if you lock the chirality at this molecule, you dictate the chirality of RNA. Okay? So it satisfies everything we want from the mechanistic standpoint, and it's the perfect molecule to work with if we want to tackle the chirality of RNA. And it's right at the center of the prebiotic chemistry. And that's what we did. We crystallized this molecule on an iron oxide surface. Another name of it is magnetite. It's the most most magnetic natural mineral on Earth, as well as the most abundant natural mineral on Earth. And when we crystallize RAO from a mixture of different stereoisomers and from its racemic state on this magnetized surface, we found that the crystals that form on the surface are highly enantioenriched. Okay? So on the left, you see the magnetite surface, and on top of it, the crystals of RAO. And on the right, you see the CD, the circular dichroism spectra of the crystals we collect. And I will walk you through what this means. So this is an optical technique to basically measure the chirality of crystals. It's using polarized light and its interaction with chiral crystals, just kind of like what Pasteur did. And it essentially, in its presence of signal, means that there is an excess in the enantiomeric excess, because when chiral molecules absorb polarized light, they absorb in a selective way, and if there's any excess, then it means that there will be an excess in the chirality. And here you see the red spectra, for instance, which corresponds to the north pole of the surface, and on the upstairs, you see the spectra, the blue one, which corresponds to the south pole of the magnet. And they basically tell us that the spin direction of the surface directly determines which enantiomer is going to be crystallized preferentially. And the black line here is a control experiment in which we used a non-magnetizable substrate like silica, still keeping the magnetic field in place, to verify that this is not a magnetic field effect, but a spin effect, which comes from the spins of the electrons. So for the case of silica, even though there is magnetic field, you wouldn't have a spin bias, and that would give a null signal, right? And the most impressive thing about the measurement here is that the numbers are quite high here, okay? Like with like physical factors that couples the chirality, like circularly polarized light or some other obscure effects, typically the excess you get are in the levels of ppm or like percent level. But here we are nearly getting unity in enantioselectivity, which is, and at room temperature in dirty conditions in kind of a solution phase chemical system, which is relevant to the origins of life. And if you essentially redo this experiment, meaning recrystallize these molecules from their enriched solutions, you can essentially get to homochirality as soon as you start from excess is about 25%. Okay? So this is kind of the threshold about which you can get to homochirality, and it is way below the average EEs we get in the first crystallization step. And what that means is in 2 crystallization steps, from 0, we can go to like hero, like homochirality. So this was the first experiment. And the second one is going to be about the about increasing the spin polarization of the surfaces. Because so in the initial one, we used an external magnet to basically fully spin polarize the surface. And that was the main reason why we got very high, enantioselectivity. And although there would be some bias in the natural environments due to Earth's field or some other factors like lightnings, they're not going to be perfect. But can we make better is the question, and now I'm going to talk about that part. So how are we going to obtain more and more highly spin polarized surfaces under natural conditions? Again, utilizing the CISS effect. And I've done these experiments at Weizmann Institute in Jerusalem. So it is the same physics, okay, that I talked about before. So it's the same thing kind of applied in the other direction. So in the previous experiment, what we had was we had a fully spin polarized surface and a mixture of molecules. Now we have a surface with a mixture of spin states, meaning a demagnetized surface, but an enantiopure solution of RAO. So now, again, because of the same spin interaction, as the molecule approaches the surface and gets charge polarized, and that charge polarization accompanies the spin polarization, the spin state that is at the end of the molecule will interact with the surface and magnetize it in the direction that is energetically favorable. So again, it's the same physics, it's just a different way of looking at it. And we probe this effect, meaning the chirality-induced magnetization of the magnetic surfaces, with a technique called magneto-optical Kerr effect microscopy, which essentially reflects a polarized light of a surface, and based on measuring its polarization change as it reflects from the surface, it gives you a contrast which reflects the magnetization. So it's a polarization contrast imaging that tells you about the magnetization differences. And in this experiment, we have a magnetic surface, and on which D or right-handed or left-handed crystals of RAO are present. And then when we apply magnetic field to this surface, we observe that the magnetic surface behaves differently in the vicinity of chiral molecules than in the absence of the chiral molecules. And I'll get more specific about what is happening and some numbers here. And what we did with this experiment is we did something called Kerr effect hysteresis measurements. And essentially, we measured the hysteresis of this material nearby the chiral molecules and away from the chiral molecules, and tried to understand how chiral molecules affect the magnetic properties of the surface. So one way to read this measurement is from here. And essentially here, we look at 2 different areas. The area that is shown by blue is the area that is free from crystals. And here, there are the chiral crystals. So the red area is kind of in the vicinity of the chiral molecules. And if you apply a large negative field, both areas are basically perfectly magnetized. And as you increase the field in the opposite direction, up to a certain extent, you can basically retain the magnetization because of the internal magnetic interactions. But at a point, the domains that are free, basically away from the chiral molecules, basically give up, and then they magnetize in the other direction, in the positive direction. But the area that is around the chiral molecules do not. And it persists more until you apply a larger field by about 1 millitesla, which is about 20 times stronger than Earth's field. What that means is chiral molecules induce an added magnetization, which is not actually because these molecules are like magnets, but because they induce some spin exchange pinning-like effect, which preserves the magnetization longer than it should be. And this has several consequences for prebiotic chemistry. One is essentially chiral molecules can induce magnetization, and the induced magnetization can be stronger, much stronger than Earth's field. So essentially the magnetization that is induced by the surface wouldn't flip even if there were some fluctuations in the environment, like for instance, the reversals of the magnetic field. But also, this effect is kind of like an avalanche crystallization, meaning here we don't directly look at the domains underneath the crystals, because actually technically you cannot directly look under it because the crystals scatter of light and you need to look a little bit nearby. But what is happening is, because of the finite size of the domains, if there are enough molecular spins in the domain, they basically flip the magnetic domain, and because of the finite domain size, the magnetization spreads in an area that is larger than the area that is covered by chiral crystals. So essentially, the molecules kind of align the spins underneath them, and then the magnetic domain, basically through magnetic interactions, aligns the spins nearby, and the effect kind of spreads like an avalanche. Okay? So it's a nonlinear effect, and that's important, right? You need some nonlinearity to amplify your spin polarization. So, I mean, you can probably see where it's going. We have like chiral molecules affecting the surface and magnetic surface affecting chiral molecules. So this can kind of be a feedback, right? Like one reinforces the other, and then the other one basically picks the right chirality. So look at this system, where we start from a completely racemic and completely depolarized system. And here I just showed, as a reminder, two effects that I described, the first one and the second one. The first one was how magnetic surfaces choose chiral molecules, and the second one was how chiral molecules induce magnetization on the surfaces. So now let's think of them together. Okay? We have a surface that has no spin bias, and a solution that has no enrichment. So one molecule of RAO will basically crystallize on the surface stochastically and induce some bias. And because of the avalanche property of the crystallization, it will spread around the magnetic domain. And now, and that will be because of effect number 2. And now, because of effect number 1, the domains that are aligned will basically perfectly favor the right-handed enantiomer, meaning it will attract those molecules faster than left-handed ones. So essentially, you will start covering this area with the right molecule, and then that will spread out because of effect number 1, and so on and so forth. Okay? So you can kind of think of these in combination with each other. And if you start from a little nudge in the bias of the magnetization that would come from Earth's field, you can amplify this and selectively crystallize molecules at the same time. So here I showed that kind of interplay. We start from a racemic mixture of the molecules, and then a rock basically that has some statistical bias that comes from Earth's field. And then that induced bias will basically induce some enantioselectivity on the molecules. And then with recrystallization, you can essentially get more highly enriched molecules. And because these are conglomerate crystals, as they grow, they kind of spread out in the domains in an enantiopure way, and then they will align the domains underneath them, so you will realize more and more spin polarized surfaces, and then the next crystallizations on the surface will be more and more enriched. So this system is able to kind of reinforce this two effects together in like a feedback. So now the other stuff that I want to talk about is how this fits into an environmental context, and it's stuff that we worked on here in Cambridge and partially at Harvard. Why are you laughing? Is that you? Okay, so we envision this, this system happening in kind of a shallow lake with maybe some stream that is carrying with a racemic stream of these molecules that were synthesized by John's chemistry. And as these molecules kind of flow over a rock surface which was magnetized by Earth's field, you kind of, almost like a, like a filtering coffee, based on their enantioselectivity, you filter out the right enantiomer in that area, and then the others will be washed away. And if you kind of think of a connected ponds, like the Paternoster lakes, you can realize a small area with enantiopure crystals where the lightly enriched supernatant solution will be carried away and diluted in the other parts of the environment. So this is kind of the rough geological scenario that I've been envisioning. So one question is how much magnetization you can get, and can we measure that in a sediment that we find in the field? So that's what we did at Harvard. And here you see Roger Fu, my colleague at Harvard, picking up some sediments from a very dirty pond in Cambridge. And because he's a geologist, he's doing the dirty work there. And when we collected some sediments and resettled them in a beaker under Earth's field, so here there is no external magnetic field, there's only Earth's magnetic field. We basically expected these sediments to have some magnetic inclusions like magnetite naturally. And as they kind of settle down under Earth's field, we expected them to have some statistical bias along Earth's field, or I guess the field in Boston. And we immobilized these sediments with some technique in epoxy and imaged them by a sensor that is sensitive to spin. And this is a technique called quantum diamond microscopy, which takes advantage of spin centers in diamonds. And it's essentially a magnetometer which works at very short length scales and can measure magnetic field fluctuations of very low strengths. So this is the sediment you see that we collected from the lake, and this is like Z upward, so this is like the water is upstairs. And when you image this sediment with magnetic resolution using NV centers in diamond, you can detect the magnetic sources. Okay, each like black spot here is a magnetic source, and this is the strength of them. And when you take the gradient of this map, you can essentially identify individual dipoles that correspond to a certain alignment of these magnetic inclusions inside the sediment under Earth's field. And I mean, one way to analyze this is staring at it long enough and see that there's a bias toward one direction, or you can use Python, and I did the latter way. And you see that these sediments indeed have a bias, and when you average them together, they basically point toward the direction of Earth in that location. And also, if you collect sediments from different areas in the same large area but different locations in the local area, they still have the same bias. And that's what these three dots show. They basically are from the same lake but from different spots. And they not only match together, meaning there's internal consistency, but they also match the field direction of the place where we did these experiments. And also you can look at the numbers and kind of calculate how much bias there is, and this is still work in progress, and we will publish these experiments very soon. But what this shows is, in a natural environment, when the particles are kind of settling down and kind of like scrambled around, there is still bias that would come from Earth's field in a natural environment. And the second thing is, which we're working on with Nick, Clancy, and Rich here, is about how would this happen on early Earth? What would be the synthesis route of magnetite or the magnetic minerals in a kind of a lake environment? And what would be the magnetic properties of these inclusions? Okay, so what Nick discovered previously in the context of Mars is that magnetite can be synthesized from a very simple route very effectively from iron. And Earth was very rich in iron. and essentially at higher pH, it precipitates as a species called white rust. And then you can selectively oxidize that species with UV light and make an intermediate called green rust, which then makes magnetite. And then that magnetite, as was discovered in Gale crater, for instance, exists as single domain inclusions in kind of a matrix, as sometimes clay, sometimes silica. But they're like kind of these single domain inclusions in that matrix, which individually they're kind of aligned under Earth's field, and each domain here acts kind of like an enantioselective catalyst because of all the things we discussed, okay? And Rich was someone who basically pointed out the importance of the magnetic domain properties. And essentially, as these particles get larger in size, they split up into domains because the material cannot hold the magnetic energy. And if the particles are too large, they basically split up into domains such that the overall magnetization is canceled. So this part is a stable, state of the inclusion, but it has low magnetization. On the lower end, when the particles are very small, the magnetization is very easy to be aligned because the particle is very flimsy, but also because it's very flimsy, it can fluctuate very rapidly. So somewhere in between is the perfect spot where like the particle is effectively in the single domain structure, meaning it has high spin polarization, and it's also not too large that it doesn't split up into multiple domains. And when Rich made these measurements, he found that actually the magnetite that we made with Clancy in Nick's lab actually correspond to the area that we wished them to be. And in this so-called pseudo-single domain regime, where there is high spin polarization and stability of the magnetic field alignment. Okay, so I talked about the mechanistic parts. I talked about how that fits into natural environment, and now I'll talk about the final part, which is how this story ends up being a homochiral network, right? Because I talked about just RAO molecule, and that's one molecule. So let's say, I hope that I convinced you that we can get this molecule homochiral, but how are we going to propagate it in the network? And I will end with this. So what would be the effective way of propagating chirality in a network, I guess, is the question. So kind of we can even forget about what we talked up till now and ask ourselves this question. There are multiple classes of molecules in prebiotic chemistry. There's nucleic acids like RNA, there's peptides, and there are metabolites. What would be the ideal class or level of molecules to basically start propagating chirality from? Okay, so we can start from metabolites. So you see here the citric acid cycle, which is the Krebs cycle. And one molecule that is relevant to this cycle is malate. And malate actually can be obtained by the reduction of tartrate, which is something that Ziwei discovered. And tartrate can be obtained in its homochiral form with the mechanistic steps that we talked about. But the interesting thing is, as malate enters the cycle, what can it do? It either oxidizes the oxaloacetate or dehydrates to fumarate, both of which are achiral molecules. Okay? So essentially, it can be made homochiral, but as soon as it enters, its chirality cannot be propagated because it's kind of in a chirality grinder, right? And it's the same thing with isocitrate. So in the absence of enzymes that are driving these processes, just the abiotic Krebs cycle is kind of a chirality grinder. And it's not an effective way to start chirality because you cannot propagate it from. Okay, so this was one argument. What about amino acids? Well, there are 20 of them. 3 of them can be crystallized as a conglomerate, like the crystallization that I showed before. But they're very soluble. It's very hard to crystallize them. And there's many of them and with idiosyncratic chemistries. So you have to basically go through and do this thing for every single of them, which is not very elegant, unless there's a better way. And now I'm going to show that. And I think the better way is to look at this experiment, which was done in Sutherland lab, which shows that if you do peptide synthesis, meaning ligating 2 amino acids together using RNA-coded peptide synthesis, you essentially get a stereoselective interaction that favorably makes L peptides using dRNA. So essentially, you can propagate chirality from RNA to peptides, and in the direction that we want, because life uses right-handed nucleic acids and left-handed peptides, and this experiment basically verifies that using tRNA analogs with coded peptide synthesis, you can propagate chirality from RNA to peptides. So let's put them together. D-tRNA makes L peptides, or vice versa, L-tRNA makes D peptides. L peptides will make L enzymes. And if the enzymes are driving the processes in the metabolic cycles, then this process will not be a chirality grinder anymore, and it will be homochiral. So essentially, you cannot propagate chirality from here to there, in the other direction, but you can propagate it from this direction. And what does that mean? It means that if we can make right-handed tRNA, we can essentially propagate that chirality first to peptides and then to metabolites. Well, what is the way to make RNA homochiral? If you make individual units, meaning ribonucleotides homochiral, you make RNA homochiral. And what is a precursor that can make all ribonucleotides homochiral? Well, I guess we know such a precursor, which is RAO, and we know how to make it homochiral. So essentially, you reduce the colossal problem of homochirality of individual units like metabolites, peptides, and nucleic acids, and all the other stuff to making one molecule homochiral from which chirality can be propagated effectively. And that's a much elegant solution, in my opinion. And we know how to make this molecule homochiral. And the direction of this process, meaning the unidirectionality, kind of resembles the direction of information flow in life that is established by Francis Crick, I guess here, which is from nucleic acids to peptides, right? So this is Crick's dogma, the sequential information that flows this way but not backwards. And the chiral dogma, that's why we call the central dogma of homochirality, also, I guess we suggest that to be, flowing from nucleic acids to peptides and then to metabolites in a unidirectional fashion. So it's nice, I guess, correspondence. And with that, I will end my presentation. And of course, I talked about a lot of things, and they're in my papers from my PhD. So if you want to take a look at the details or discuss, please do so. I would like that. And I also thank my collaborators, my PhD advisor, Dimitar, colleagues who do chemistry incredibly well. Most of them are here. And my collaborators who discovered the CISS effects, they're in Israel, and the collaborators in Cambridge here. And thank you. And I'll leave with my summary slides, so maybe you want to take pictures.